Hello, I'm Steve Mao, Senior Editor of Science. Today, I'm very happy to be here to have the opportunity to interview Dr. Lingling Chen. Lingling is a professor and principal investigator at the Shanghai Institute of Biochemistry and Cell Biology of Chinese Academy of Sciences. And although Lingling set up her independent laboratory less than 10 years ago, she has been a veteran in the RNA research field. She has worked on allo repeats, derived RNAs, link RNAs, circular RNAs, and precursor RNAs that generate small RNAs. So Lingling, it's nice to be here with you.
Thanks, Steve.
So as I said, you have been studying RNA for a long time. So in this last, I guess, 2 decades or so, what do you think of the progress made in the RNA field?
2 decades, that means about 20 years ago from now. Let's count. It's gonna start from 1990s. So I think we have the wave of the non-coding RNA being discovered by so many different labs, from small RNAs to long non-coding RNAs to circular RNAs. Those molecules are so fascinating. I think the discovery of so many non-coding RNAs has been changing our view of look at how genes are regulated, how those non-coding RNAs can impact cells in many ways. So it's been very lucky to work in RNA.
Yeah, so as you said, it's really the so-called junk portion of the genome, right? People used to speculate those are junk portion because they do not code any proteins, but now it turned out that they actually code very important RNA species, and many of them play various roles, actually, which is the main theme of the meeting this year. As I know that you started with link RNA, and later you moved towards a precursor RNA that generates small RNA. So can you tell us what made you switch from a link RNA to study this particular precursor RNA?
Well, I wouldn't call it a switch because it's really a smooth discovery. I would say back to 10 years ago, one of the most exciting discovery in molecular biology, in my opinion, was the pervasive transcription of the genome, leading to the discovery of those intergenic region transcribed non-coding RNAs. Now we know that this transcription and the processing of non-coding RNAs are different from those of mRNAs. But at the end, those mature link RNAs look similar to mRNAs. They have 5'7G and 3' poly tails, with only a few exceptions. Those NIT1, MALO1, originally discovered by David Spector lab right here at Cushman Harbor lab. So his lab found that the 3' processing of these two basically use the RNA cleavage related with tRNA biogenesis. And those RNA look so different at the 3' end from mRNAs. And I luckily worked with NIT1 with Gordon Carmichael. At the time, originating from those allo elements, I found those mRNAs contain inverted allo repeats is preferentially retained in the nuclear body called paraspicles. And I found NIT1 is the major organizer of paraspicles. So triggered by the very interesting different appearance of NIT1, I asked the question, do all the RNAs look the same as mRNAs? So from there, I began to explore the non-poly A transcriptome, which has been ignored by so many people, so it's just junk. So looking into the non-poly transcriptomes, we discovered so many different, well, basically 4 different classes of previously unknown species. These RNAs do not have their own promoters, but are rather processed from the primary POL2 transcripts and then stabilized by distinct mechanisms by forming RNA circles, by the protection of small RNA protein complexes at each end or on one end. And more importantly, now we figure out that some of them can impact those important functions of gene regulation in cells, also related with human diseases, including Prader-Willi syndrome and autoimmune disease like lupus. So we're happy to see those previously thought the junk thing can do something in cells.
Yeah, so that's fascinating. Yeah, as you said, you started as a molecular biologist to study the cellular phenotypes of those RNAs, the retention of the ago-containing repeats, and then you move towards a poly-A minus portion, and then you discover this human disease-related small RNA precursor, and you also mentioned the circular RNA. Now you have worked on the role of circular RNA in lupus. I just want to ask you, so what is it like to transition from molecular biologist to all these different fields? I know that you studied immunology in the lupus setting, and also you studied human embryonic stem cell, basically the differentiation potential, the function of those RNA. So can you say something about that?
Well, I think it's been very challenging for me. As you know, I was trained as an RNA biologist. So all the RNA stuff from my lab was rooted in the molecular biology. But we just take all or every approach necessary to address the fundamental question I want to ask. So for example, my lab is routinely using the imaging as one way to look at how exactly a particular link RNA localized in the cell. They're going to provide lots of information to predict what they do there. So if happens to be like we found those RNA circles because they are slowly generated and fastly degraded, and when they are there, they form those very interesting, imperfect, intramolecular double structures that can prevent unwanted double RNA binding protein, which is also immune factor like PKR activation that's related with autoimmune disease lupus. So if it happens to be autoimmune disease, so we just do collaboration. For example, we got those precious SLE patient samples from the local collaborator, like Dr. Nanshen from Renji Hospital. So for this something, because I really don't know immunology, so we can do collaboration with colleagues. But for all the other things, we try to do the most we can in the lab. So I just feel like, as I said in the beginning, we try every approach necessary. The ultimate goal is we wanted to address what those mystery RNAs do in cells.
I see. I really like that. I think the ESO is really to take whatever approaches and go into whatever context to address the fundamental question you want to answer, which is what is the biological function of those non-coding RNAs. I think you were being modest saying that you do not know immunology. I think quite to the contrary, and at this meeting you just showed a very beautiful story.
Thank you.
So to summarize, basically, Lingling found a group of circular RNAs, and they are fastly degraded by RNase L upon infection. And she showed that those circular RNAs actually sequester and very important innate immune sensor, protein kinase R, PKR, because they have these special structures. So upon infection, those circular RNA were degraded. So PKR is now released to presumably to function as an innate immune sensor. So I think this is a very fascinating story that linking molecular biology, fundamental question in molecular biology, what's the role of the circular RNA, with very important disease-relevant role of innate immunity. So you mentioned in your talk, at the end of your talk, that it might provide actually a direction for therapy in the future to treat lupus patients. So can you elaborate a little bit, like how we can use circular RNA to to kind of advance into the clinics.
Thank you so much for bringing this point. So we were actually very excited to see this. For example, during our experiment, if we introduce those iron circles, because normally those lupus patients, they have global reduction of circle expression accompanied with aberrant PCAR activation. So now if we introduce iron circles into the blood cells came from the SLE donor, we're seeing the reduced PCAR activation together with this the downstream cascade, including interferon responses, interferon-induced genes, and many of those genes have been used as a diagnostic marker of SLE. So it's really suggesting if in the future we can introduce the in vitro synthesized circles without immunogenicity to, at least for example, in the beginning, the animal models, we would like to see the circles can reduce those unwanted PCAR activation that are going to be benefited to the patient, hopefully in the future.
Yeah. I see. Yeah, I think that would be very exciting because in this meeting we've seen that actually a lot of this RNA has been moved into the clinics. For example, the Spinraza to treat SMA is an antisense oligo to block the splicing. I think it will be as fascinating if you could advance the circular RNA therapy into the clinics. So we talk about, because of the completion of the human genome, we started to understand the non-coding portion and the importance of that and the function of that and the regulation of that. But at the same time, in the last 10, 20 years, we've also seen kind of the revolution of all those biotechnologies. In your talk, you also described a very promising technology to try to study circular RNA. So can you tell us something about that?
Yeah. So basically, I would say the biggest challenge in the circular RNA field was those iron circles. They have the structure conformation. They have almost the complete same sequences with the linear RNA that basically from the same parental genes. The only difference that is back splicing junction side. So because they normally came from the middle exons of the coding gene, so make it very hard to interpret the circular RNAs only without affecting the linear RNA expression with current existing CRISPR-Cas9 genome editing type of technology as well as the RNA, because any half of the RNA also targeting the linear RNA. So a couple of years ago, Feng Zhang's lab described the exciting CRISPR-Cas13 system. So this system can be programmable to target RNA with high specificity. And one of the very unique feature of this system is intolerant to mismatch that hybridizes to the target RNA. So we were triggered by this discovery. I wanted to ask whether this technology can be used to only targeting and degrade the circular RNA without affecting linear expression. So we did a panel of this screening, and we found one of them called Cas13D from one of the family of the vast majority of the family. I'm sure maybe other Cas13 protein can also do the same thing, can discriminate circular RNAs from mRNAs. And importantly, we also can manage this system to apply the larger scale functional circular RNA screening. So that means now with this new tool that added to the existing toolkits, so we should be able to study individual circular function, not only at the individual level, but also at larger scale levels in different biological settings. So I'm really very exciting. And hopefully in the future, we're going to see more individual circular function can be discovered by different labs.
Yeah, that's very exciting. And then you also mentioned that that actually enables you to screen. I think that's also a very powerful approach. So actually a lot of your major discoveries come from this kind of non-hypothesis driven genome-wide kind of discovery. You mentioned the poly A minus portion, and then now you started to do the screen. And I think you connect this kind of hypothesis-free data set with hypothesis driven project very well, because you first find the candidates and then you set up the hypothesis and then you start to test it. I think this is a very good approach. But the challenge of doing this kind of data-driven research is that not every biologist is trained as a computational biologist. So you really need to have the kind of expertise to analyze the data. So I know that you have a very wonderful collaborator who's a computational biologist. And so can you say something about your experience collaborating with computational biologists?
Well, I think I'm super lucky because my collaborator, Dr. Yang, currently he is also the principal investigator at CAS, called PSCB Computational Institute at the PSCB. He also is my husband, so we have lots of discussion, even when we're taking care of our daughter. So I think my suggestion to do this kind of collaboration is to do discussion with your collaborator as much as possible, because in this way you can be fully deliver what kind of the key question you want to address. So that's it. I really appreciate it.
Yeah, so this is actually great advice, and I think for any kind of collaborations, this open, candid dialogue is very important. But I'm sure
we have more opportunities to talk.
Yes, of course. Yeah, not every scientist can have that kind of opportunity. The last question is not necessarily related to what your research. It's really my observation, because I had the privilege to visit a lot of universities and institutes all over the world. So I have done many lab visits in China. So I've noticed that there is a big gender imbalance. Although I understand that a lot of institution leaders, university presidents, department chairmen started to realize the issue, and they have planned to tackle that. But at least for now, from what I see, that there are really not a lot of female scientists, especially at the principal investigator level in China. So I just want to ask you, so as a female scientist in this kind of male-dominant setting, what's the biggest challenge, and what things that you think could be done to kind of improve that?
Well, it's true. So we have one number that's really agree with your observation. For example, now in my institute, we have, when the students got the PhD, you have at least 50 female, 50 male students.
You mean 50%, right?
50%,
yeah.
Right. But at the PI level, principal investigator level, we have only 20% as maximum. So I think one of the reasons is many female scientists, they were discouraged by those apparent difficulties when they want to have family. So here I have one advice for the female scientists who want to have family. I think don't be discouraged by this kind of obvious or apparent difficulties. Just ask for help, ask for support, to build up the support network with your families, with your friends, to pass over the first several years when the kids are young that needs a lot of intense attention, and after that, the life is life. But in the meantime, I always encourage female scientists to just be self-motivated, be self-confident, and persistent. If you love science, give it passion. And so in this way, maybe 10 years later, you're not going to regret, oh, I gave up my science. So just do not be discouraged by those apparent difficulties as a female scientist. That's my advice.
I see, that's great advice. Yeah, thank you so much. Yeah, it's a pleasure to interview you.
Thank you, Steve. It's my great honor too.
Thank you.